Assalamualaikum and a very good day I'll be to Semio and all my friends. I am Nuris Alaina binti Kamaruzaman. My group will do a presentation about DNA extraction. Firstly, we move on to the introduction of this presentation. What is the DNA extraction? DNA extraction is actually a procedure used to isolate the DNA from the nucleus of the cell. History started when Swiss physician named Friedrich Miescher did the DNA extraction in the laboratory. Extracted DNA is really helpful to carry out the experiment for molecular analysis, electrophoresis, sequencing, fingerprinting, and also cloning. We are familiar with those words, but electrophoresis is actually a technique used to isolate the DNA, RNA, also protein molecules according to their size and also electrical charges.
Result and observation. At the end of the experiment, we were able to see the strawberry DNA, which will appear as gooey clear or white stringy stuff. This can be seen after the alcohol is added. Next, so where exactly the DNA can be seen? The DNA can be seen in the layer of alcohol. Next is the function of each apparatus or the materials that is used during the experiment. First, detergent. Detergent will help to lyse the strawberry cells and release the DNA into the solution. Next, salt. The salt will help to create an environment where the different DNA strands could gather and clump. We also can see bubbles, which is because of the detergent. Next, alcohol. After the cool rubbing alcohol is added, the alcohol will precipitate the DNA out of the liquid, while the rest of the liquid remain in the solution. Question. So the first one, why is the strawberry mixture processed through the blender? We process the strawberry mixture through the blender because we want to break down the cell wall, the cell membrane, and nuclear membrane. From this, the DNA of the strawberry can easily goes out of the strawberry liquid. The second question, why do we need to put the mixture in ice water bath? Ice water bath will help to protect the DNA by slowing down the enzyme that can break it apart.
Hi sir, I am Tamendi Subramaniam from Kulia 7. Today I would like to explain about DNA extraction, which is discussion. So DNA mostly in the layer with the alcohol because the cell's DNA is usually protected from such enzymes called DNAases by the nuclear membrane. When adding detergent destroys that membrane, the cold alcohol helps the DNA precipitate more quickly. Salty water helps the DNA precipitate, solidify and appear when alcohol is added. Possible errors that occurred during experimentation include different filtration methods, drying methods for mashing the strawberry, and the temperature of the rubbing alcohol. If repeated, more samples would be tested. Cheesecloth would consistently be used as the method of filtration, and the rubbing alcohol would remain in the freezer up until it was needed. Finally, the conclusion. This hypothesis was supported by the experiment because at the very end of the experiment, a hook was able to pull out of a test tube a long strand of DNA molecules. This proves that DNA can be extracted from any kinds of cell. Human bodies have DNA just like a strawberry. Obviously, humans aren't strawberry, but strawberry and human DNA have a similar structure. That's all from me. Thank you.